First of all, I want to thank everybody on the committee for inviting me to come and speak on part of my dissertation work. Hopefully I can fill the full 40 minutes, but there's a lot of information, but I probably will fly through it. So, amphibian decline all over the world is becoming of increasing concern. There are many factors that influence amphibian decline, including climate change, invasive species, pollution, pet trade, disease, and a lot of these stem from one of the major problems for any animal species, habitat loss. Some of the management issues with habitat loss and some of these declines include habitat use and quality. So we need to know what type of habitat these organisms are using, the quality of that habitat. You can have forest all over the place, but if it's a certain type of forest, like planted white pine, it might not be an optimal condition for species such as salamanders. We need to understand the movement of these organisms. Are they moving from pond to pond? Are they staying at one location? How are these ponds connected? And what type of protection should we put around wetlands and the upland habitat surrounding the wetlands? We can approach these using a number of techniques, including population genetics, which is what I'm going to talk about today, and some other types of management or mitigation includes relocating animals, repatriating them, or reintroducing them into populations, and translocation. So using population genetics, we can look at how these animals are connected and come up with proper management plans. The eastern tiger salamander is one of the species you hear a lot about here on Long Island. It's within the Amphibian Decline is the scientific name. It is listed as endangered in New York State. They are decreasing throughout their entire range, and as we all know, many areas of Long Island are in increasing danger of more and more development. Currently, the eastern tiger salamander is found along the East Coast, down into Florida, up through the Midwest, and then a disjunct population is in eastern Texas. Recently, the other former subspecies for the Amphibian Decline have been separated into their own species. So the western species are no longer tigrinum. I think there's 2 or 3 different new species that have been split off from this complex. Historically, in New York, the tiger salamander ranged all the way up through the Hudson Valley. The red spot there was a historic specimen. It was a visual encounter by somebody back in, I believe, the 1800s, but a lot of people seem to think that it was probably a misidentified spotted salamander, but it's still in the records. And as you can see, historically, Long Island was just perfect habitat. Salamanders were everywhere. Currently, this is from the New York Herp Atlas. They are only found in a few ponds here on Long Island. We believe that they've probably been extirpated or removed from Nassau County. So when I was doing surveys, I wasn't able to find any in some of the historic ponds. Some of the historic ponds aren't even there anymore. And the stronghold is here central, in the central Pine Barrens region here. And then there are a couple small populations out in the Hamptons. However, we weren't able to collect a lot of samples from that region. So I'm mainly concentrating my work here in central, around here, Brookhaven region. Currently, legal protection for this species, they are listed as state endangered, but the protection for their habitat is listed under the Freshwater Wetland Act. So the actual protection is for the wetland, and the buffer zones are for the ponds. And as long as the wetland qualifies as a protected wetland, the current distance is a 30-meter buffer surrounding the wetland. 30 meters really isn't that big. It's about 100 feet. However, for tiger salamanders, the DEC can recommend that a buffer zone be 164 meters, so that's about 1.5 football fields, and no more than 50% of the upland habitat within 300 meters of a breeding pond can be converted to unusable habitat. And this is based on a single study that took several salamander species, clumped them together, and made an estimate of what a buffer size around a pond should be. But keep in mind, 300 meters, 3 football fields, that's really not a lot of space. And it doesn't say what type of habitat to preserve. We don't know how these ponds are connected, so there's a lot of questions out there on how to legally protect these species. So, since this is Long Island natural history, I'm going to throw in some Long Island history. Up until the 20s or 30s, Long Island was mostly farmland and weekend homes for the wealthy. And then in the 20s and 30s, we had an increase in tourism, and a lot of the state parks and wildlife refuges were constructed. And keep in mind, state parks and refuges back then were not managed the way they are today. And then in the 1960s, suburban sprawl, major increase in development. So in a very short time, we see a lot of rapid habitat fragmentation. And of course today, there's well over 7 million people on this tiny little island of under 1,500 miles square. So there's a lot of people, a lot of development, a lot of roads, a lot of fragmentation. Fortunately, the Pine Barrens region has been fairly well preserved. Many areas still have minimally disturbed habitat. The Pine Barrens boast a very diverse pitch pine, oak forest type habitat with lots of cool understory vegetation and lots of little wetlands dispersed throughout there. And we're lucky enough here to have many federal, state, and private protected lands. So I'm getting back to the connectivity because this is where the genetics come in. Fragmented landscapes resulting from anthropogenic or human habitat modification can really impact the dispersal, the gene flow, and persistence of any wildlife population. And if you reduce that genetic variation, if you reduce the number of organisms, reduce the number of individuals that can move from one place to another, it can affect the ability for a population as a whole to respond to some kind of environmental change, some disease, or some kind of crash, like a bad winter or a hurricane. So the two main goals of my study were to assess the population genetic diversity. So just look at how diverse the population is in general for the tiger salamander populations here on Long Island. And I also chose a few ponds in New Jersey. They're also protected there. Habitats are similar, though Jersey has a few more coastal tiger ponds. But it also gives a good outgroup to compare the Long Island animals to. And then quantify the genetic and landscape connectivity among the ponds to identify if there are potential corridors and the availability for these animals to migrate from one pond to another. So I was able to collect samples from breeding sites, 17 ponds on Long Island and 9 sites in New Jersey. There were a few sites on Long Island I really wanted to get to, but that was at the time where they were trying to build that water park thing in Riverhead, and I couldn't get to the ponds there. And New Jersey sites were pretty spread out through the southern part of New Jersey. I tried to collect as many samples as possible, and as many of you might know, salamanders are very difficult to find, and you just got to be on the spot, the right time, right place to get a sufficient number of samples. So some ponds I was only able to get 2, and then luckily I had several ponds here on site that I was able to get up to 93 samples. So big range of different samples. And then I just used a small tissue sample. Early on I used a small part of a toe, and then some tail tips, and then individual eggs from egg masses. So I didn't want to disturb entire egg masses, but I was able to take one little individual from egg masses from time to time. So this is some genetic garbly stuff. Basically, we extracted the DNA from the samples, and we were using microsatellites. Now, microsatellites are short repeats within the genetic code that can be unique to individuals, can be unique to different groups, And it's a very easy way to look at very recent changes in genetic diversity. And then this is just the equipment I had to use at Cornell. It's pretty fun stuff. It's really neat to see this stuff work. So I genotyped, or I looked at 439 individuals across 12 different microsatellite loci, and these are 12 different parts within a specific part of the genome. One of the things we wanted to look at, if there were alleles, the null alleles are basically ones that aren't really coding for anything. They don't help in the analysis, so you want to, if they're not going to work, you want to get those out of the analysis because they can change your data. I didn't see any linkage disequilibrium. That's another one of those things where you, if you have problems with that, you'd have to throw out those data set as well. And most of the individuals in the ponds conform to Hardy-Weinberg equilibrium, which is another comparison to make sure your data are analyzable without any kind of bias. Overall, we saw very low allelic diversity. So all the animals were very similar genetically. Most of the markers were not very polymorphic, so there were not really a lot of diversity at each of the sites. So we only saw 1 to 3, on average, 1 to 3 different markers within our, alleles within our markers in New York, and then New Jersey was even worse with 1.7 to 2.4. So really, really low diversity. For example, using a similar marker at Purdue, a colleague of mine was able to find 64 alleles at one marker. So their diversity is a lot higher than what we have here. So this chart's a little crazy. This basically shows the patterns of allelic richness. And the dark gray bars here are the average number of alleles that you find at each pond. So these are just the New York ponds, 1 through 17. So here, at pond number 1, the average number of alleles was about 3, which compared to the other ponds, that's okay, but it's still not very diverse. The medium ones are the number of effective alleles, so these are the ones that are actually being used in our analyses. And then the white bars are the number of private alleles, or alleles that just show up at that particular site only. So there aren't too many private alleles, but there are a few. The important part of this one here is this bar up here. This shows the average heterozygosity. Now, heterozygosity is of major interest when it comes to genetic variation in natural populations. It can tell a great deal about the genetic structure and the history of a population. For example, species with low natural heterozygosities like cheetahs and black-footed ferrets, if you've kept up on any of the research on those, they're very low diversity within those groups. And it shows that there can be severe effects of small population sizes that could have been caused by a population bottleneck, a large number of individuals dying off and then starting again from a smaller population, building back up. A high heterozygosity, which it ranges from 0 to 1, so 1 would be high heterozygosity, would indicate lots of genetic variability, so lots of diversity. And then anything, so you see here, this is on this axis here, the heterozygosity rarely goes above 0.4. So that's showing that there is fairly low heterozygosity, and that means little genetic variability. If the observed heterozygosity is lower than expected, we want to look to see if we can attribute this discrepancy to forces like inbreeding or population isolation. And this is the New Jersey population as well, so same thing. very low heterozygosity, very few alleles per pond as well, so very, very little diversity. So this chart here, another really busy one, shows relatedness. So this kind of is a sort of like a measure of inbreeding. The higher the relatedness value, so the closer you get to this within these two bars. Let me start over here. So it ranges from -1 to 1. If you're 1, you're I lost my train of thought here. Okay, siblings have a relatedness of 0.5. So if the higher you get, the closer you get to 1, the more related you are. The farther away you get, the least related you are. So here we have a fairly decent amount of diversity, but there's still quite a bit of high relatedness in some of these populations. So we're seeing, this is indicating a little bit of inbreeding, but in a salamander populations, this can be expected because the amount of dispersal is typically not as often as like a mammal that can actually move from one place to another easier. And then we wanted to look at how we group these, the New Jersey and the New York populations together. So we ran some crazy statistics, and we came up with 3 distinct genetic, what we call demes, or genetic groups. The blue indicates the main Long Island group. The orange indicates the New Jersey population. And then we have this yellow here. And these 2 ponds right here in the middle of Long Island that kind of stand out. So that's a 3rd group. And so we wanted to step back and kind of look at that and see, well, what's causing this group to show this difference? And I'll get to that on the discussion part. But overall, we saw high levels of differentiation between New York and New Jersey. So New York's its own distinct group. New Jersey is its own distinct group, with the exception of those 2 little weird ones here in Long Island. Another weird thing that we've noticed is that we did some assignment tests to see if an individual would be assigned to the pond where we think it came from. And a lot of these animals were actually assigned to different ponds. So that's another indication of lower diversity. All these ponds are still fairly genetically similar, but that also indicates that we have very few alleles to work with, so it could mean several different things. Now, we wanted to look at, the second part, we wanted to look at barriers to movement between ponds. So individuals could possibly migrate from one pond to another, or if they're completely blocked from each other. So we used some GIS software and defined land cover resistance values based on a couple papers that have done work on salamanders before. And we calculated the Euclidean distance, or the straight line distance between ponds, and then the surface resistance based on the land cover. So, for example, a forested area would have a low resistance because salamanders typically can move through forests, but a impermeable structure like a building or a large parking lot would have a high resistance value because a salamander typically wouldn't make it through, or actually wouldn't even try to go across a parking lot like that. So we assigned all these values to different types of land cover, so forests, wetlands, good optimal habitat were rated with a low resistance value, and things like saltwater marshes, urban areas, that kind of thing were rated high. And then we correlated these values with some other relatedness tests. So the interesting thing is, when we did the GIS analysis, both on Long Island, you see down here, and in New Jersey, this green here indicates that a lot of these ponds actually have very little resistance between each other. So there is still the availability of individuals to go from one pond to another. There is a little concern here in this region here of Long Island. There's a canal here and a lot of development. So these ponds are very much isolated from the main population over here. But the good thing is most of the breeding ponds throughout Long Island, at least this region from Brookhaven east, there is the chance that individuals can move from pond to pond. But I lost a few things because of the Mac to PC conversion here. But we didn't see any relationship between the differences, the genetic differences between the ponds and the connectivity. So we would expect that the individuals that are close together to be more related to than the ones that are found like here in Long Island all the way out to Southampton. So there was no relationship, which is kind of interesting because we would expect the farther away they are from each other, the more differences you would observe. So bringing all this stuff together, we came to several conclusions. The low allelic diversity, as you saw on the original map, we're the northernmost part of the tiger salamander range. So this is very likely due to an initial migration during the Pleistocene. After the glaciers started receding, animals started moving north again. So the animals that were able to eventually, over a long period of time, move back up here. There were just a few, and that's how the population grew from there. But this also could make the entire population susceptible to a collapse in case of a catastrophic event. For example, New Jersey has lost 2 tiger salamander ponds thanks to Sandy because they became inundated with salt water. At least they lost 1, maybe 2 years of salamander breeding until the ponds dry up and refill with fresh water. But with sea level rise, they probably will lose several of those ponds anyway. So the remaining part of this population, if there is gene flow between these ponds, may actually lose some important genetic material from moving back and forth. And there is a very high risk of inbreeding depression if these ponds become more isolated from each other. So if these corridors that are actually existing no longer exist, the populations could really suffer and the animals wouldn't be able to disperse like they are now. So there are 3 distinct genetic demes. There is some evidence of admixture. You saw some of the orange within the blue and some of the blue within the orange. That could probably just be historic shared alleles, shared diversity, ancestral polymorphism, rather than gene flow between New York and New Jersey, because unless salamanders somehow evolved to swim across salt water, it's not going to happen. So it's probably just some kind of ancestral result. The weird group in New York, I sampled this pond shortly after some remediation in that pond. And so I'm guessing that it was a very small founder type situation where the population might have been depleted a little bit, or individuals went someplace else to breed, and then I just got a very, very small sample. And I'd like to check in 10, 15 years and see if that pond actually comes back to be similar to the other ones those ponds to be similar to the ones surrounding it. So it's likely caused by the disturbance. But that being said, that's also cause for alarm, because if a small short-term disturbance like that causes the genetics to shift that much that quickly, and there aren't surrounding individuals to come back and repopulate that, that can cause some major inbreeding problems as well and can be cause for concern. So there is some evidence of recent migration between ponds. The differences between them is likely due to distance between ponds, even though there's no definitive relationship. The distance between them, you still can't have one animal going from Brookhaven National Lab to Southampton. So you have to have some stepping stone effect there. And then corridors appear to be present both in New York and New Jersey, but there really isn't a relationship between the individual pond relationships. So this is bringing back to this map here. You've got some really nice, this is Brookhaven Lab right here, really nice connectivity. Individuals can go from place to place, but there really aren't any available corridors over here where genes can flow back and forth. So 15, 20, 30 years from now, we might see another distinct theme in the Hamptons if those populations persist, pop up. And these, some of the ponds down here in New Jersey were some of the ones that got inundated with salt water from Sandy. So some of the management questions and concerns and that came out of this, we really do need to be able to calculate buffer zones or zones of protection around upland, wetland upland habitats, and really determine them on a case-by-case basis. These areas need to be connected to other wetlands or have some kind of corridor between these wetlands to be able to protect them and allow for dispersal. We want to also estimate the probable dispersal habitat and determine the available corridors. Another part of my study was using radiotelemetry to determine movement patterns of these salamanders, and 100% of the animals utilized the pitch pine oak forest. They did not use any planted white pine, they didn't use open fields. They used every single animal bedded in the typical Pine Barrens pitch pine oak forest. So if we're going to maintain forested corridors, the recommendation should be the pitch pine oak. And then individual breeding